Hello everyone, today we are going to start with a new topic, and that is anemia. So let us see what anemia is. Anemia is defined as reduction in total number of circulating erythrocytes or a decrease in your quality or quantity of hemoglobin. Okay, so this is your normal definition. And the causes of anemia, can either be altered production of erythrocytes. Normally when we talk about production of erythrocytes, and in case of anemia there is actually reduced production of erythrocytes. Blood loss. Why blood loss will lead to anemia? Because whenever there is loss of blood, there is going to be loss of RBC as well, so that can lead to anemia. And thirdly, there is increased erythrocyte destruction. We all know that your production of your RBC is always equal to your destruction of your RBC. So your number of RBC in your blood is always maintained. So whenever this balance is broken off, in this case because of increased amount of destruction, it will lead to anemia. And lastly, it can be a combination of all these three effects. Okay, so let us move on to your next slide. So this is your general diagram of your red blood cell. Your main function of your RBC is to carry blood and to carry oxygen, and then to supply oxygen to different tissues of your body. Normally this binding of your RBC to your oxygen is carried out by hemoglobin. Okay, hemoglobin is a part of your blood in which 95 to 98% of your oxygen goes and binds to. Okay, it goes and binds to, and hemoglobin it contains a pair of alpha globulin chain and a pair of beta globin chain. So alpha, there is alpha 1 and alpha 2, so it is a pair of alpha globin chain, and there is a beta 1 and beta 2, which is a pair of beta globin chain. And these chains are mostly, they are all polypeptides, which means they are made up of amino acids. All of these four chains, at the center of these chains there is a presence of your heme moiety. There is a presence of heme. So this is your structure of your heme. And whenever we talk about your heme, in the center of your heme, again it contains an atom of iron. So this iron, it goes and it binds to your oxygen, and thereby your RBC is able to carry carry oxygen throughout your body. Okay, so your symptoms that you see in anemia basically depends upon four main factors. So the factors that determines the symptoms that you see normally are, in case of number one, it is the speed of onset of anemia. More rapid your onset of anemia is, more symptoms you are going to see. If your onset of anemia is slow, your lesser your amount of symptoms that you see. Okay, that is in case of speed of onset. More rapid your onset, more symptoms. Less rapid your onset, less symptoms. Second is severity of anemia. Whenever we talk about severity of anemia, more severe your anemia, more symptoms. Less severe or mild anemia, lesser your signs and symptoms. Okay, so in case of your age of your patient, it has been seen that younger patients, they tolerate anemia more than in case of older people. This is basically because your circulatory system of your young individual is far more better than that of your older people, so they can tolerate anemia far more better. Hemoglobin dissociation curve. So whenever there is any functional abnormality in your hemoglobin, your oxygen that binds to your hemoglobin, they dissociate far more easily than in case of normal hemoglobin. So in this case your curve tends to go towards your righter side. Okay, whenever we talk about anemia, okay, there are different types of anemia, and anemia is classified into different categories, which is determined by changes which affect generally your shape of your shape of your RBC, your size of your RBC, and your substances that is present inside your RBC. Okay, so classification of your anemia is basically done based on your changes which affect shape, which affects your size and your content of your RBC. Okay, so based on this, there are different types of anemia. Okay, so we move on to, so for your first type of anemia, which is your sickle cell anemia. Sickle cell anemia is a hereditary disorder. Okay, it is a hereditary disorder which is generally caused by point mutation. So, and it is one of your most common types of hemoglobinopathies. Hemoglobinopathies are nothing but a group of hereditary disorder. They are a group of hereditary disorder which is caused by inherited mutation. So this mutation is passed on from your parent to your offspring, and this mutation generally leads to abnormalities in your hemoglobin. So whenever we talk about mutation, what kind of mutation is seen? See, okay, there is a mutation. So what kind of mutation is seen in case of your sickle cell anemia? The kind of mutation that you see is point mutation. Okay, and because of this point mutation which occurs, you see abnormalities in your structure of your hemoglobin. So due to your abnormalities or which occurs in your hemoglobin, there is presence of sickle hemoglobin in your red blood cell. Okay, it is a hereditary disorder which is caused by point mutation. Because of this point mutation, you get abnormal hemoglobin, and this abnormal hemoglobin is known as your sickle hemoglobin, or it is denoted by HbS. Okay, we see mutation in, and generally this mutation occurs in your beta globin chain and not in your alpha chain. Okay, it affects your beta globin and not your alpha chain. So whenever we talk about your RBC, okay, your adult RBC contains 96% of HbA, which is your adult hemoglobin. Okay, 3% is HbA2 and 1% is fetal hemoglobin. Okay, see your, if you see your globin chains, they differ in each and every hemoglobin, different types of hemoglobin. HbA contains a pair of alpha and a pair of beta. HbA2 contains a pair of alpha and a pair of delta, whereas fetal hemoglobin, that is HbF, contains a pair of alpha and a pair of gamma chains. So there is difference in your different types of chains, and your adult hemoglobin contains 3 different types of hemoglobin, that is HbA2, HbA2 and HbF. Okay, I hope it is clear till here. So I said sickle cell anemia is a hereditary disorder which causes point mutation. So because of this point mutation, normally your beta chain, under normal condition, okay, your beta chain of your hemoglobin, okay, on its 6th amino acid, it contains glutamic acid. Okay, beta globin chain is a polypeptide, so it is made up of numerous amount of amino acid. A normal beta chain or your beta globulin, on its 6th amino acid, there is presence of your glutamic acid. But in case of sickle cell anemia, due to point mutation, this glutamic acid is replaced by valine, okay, another amino acid. Under normal condition, your 6th amino acid would be glutamic acid, but now you have a point mutation. Due to this point mutation, your glutamic acid is replaced by valine. Okay, so because of this change in the 6th amino acid, you get your HbS, which is your sickle hemoglobin. Now your difference in this case is, what happens is, whenever this HbS is deoxygenated, okay, your hemoglobin, they contain oxygen. When they bind to oxygen, they are oxygenated hemoglobin. When they give off oxygen, they become deoxygenated hemoglobin. So under deoxygenation, this HbS, that is your sickle hemoglobin, due to your presence of your valine, what happens is, on deoxygenation, they form long polymers. Okay, they form long polymers which are normally semi-solid gels. Okay, and then your hemoglobins, they interact with one another. They come and they stick to one another. Your RBC is, your hemoglobin is deoxygenated hemoglobin is there. Now due to deoxygenation, your HBS, they form long polymers, okay, which are semi-solid gels in nature, and then they interact with one another, they stick to one another, and then they change your structure of your RBC, okay. So they change your structure, and then your RBC, which is normally biconcave in shape, due to formation of your polymers, they change into a sickle cell. Okay, this is your sickle cell. Because of your formation of your sickle cell, when it is deoxygenated, initially this sickle cell formation is reversible in nature on oxygenation. See, it gets oxygenated, it goes back into forming a normal biconcave RBC. But whenever there is oxygenation and deoxygenation taking place continuously, okay, there will be change in your structure. Okay, what happens due to continuous oxygenation and deoxygenation of your RBC? What generally happens is there is going to be influx of calcium ions. Okay, normally biconcave, deoxygenated, it became sickle cell, again oxygenated, it became biconcave. So this process keeps on going on due to tremendous amount of deoxygenation and oxygenation taking place. Changes start to occur in your RBC. There is going to be influx of calcium taking place. So whenever influx of calcium takes place, your potassium and your water ions, water molecule, they come out from your RBC. Due to this, due to influx of your calcium and efflux of your potassium and your water from your RBC, what happens is this is going to damage your membrane of your RBC. Due to your damage to your membrane of your RBC, generally now what you get is you get a permanent sickle cell RBC, and this permanent sickle cell RBC, they, due to your damage to your membrane of your RBC, they undergo lysis, so they break off. What happened? I'm sorry. So they break off. Okay, sorry for this. Okay, they break off. So this is what happens in case of your sickle cell anemia. Okay, now there are three most important factors which affect sickling of your RBC. Okay, first is your presence of hemoglobin other than HBS. Okay, before we go into this, what I would like to talk to you about is your HBS in a person may either be heterozygotes or it may either be homozygotes. Okay, a person may either be heterozygote or it may either be homozygote. Heterozygote means in which there is only one defective allele. In case of homozygote, there will be both your genes will be defective. In hetero, one set of gene will be normal, the other set will be non-normal gene. But in case of homozygote, both your alleles will be defective. Okay, so your presence of hemoglobin other than HBS. Now it has been seen that in case of homozygotes, okay, in case of heterozygotes, it has been seen that 40% of your hemoglobin is sickle cell hemoglobin, whereas the remainder is a normal HbA, which is your adult hemoglobin. Okay, in case of heterozygotes, 40% will be HbS, which is your sickle cell hemoglobin, and your remainder is going to be HbA. And it has been seen that your HbA, okay, that is your normal hemoglobin, which a pair of alpha and a pair of beta globin, they interact very weakly with HbS. Your interaction with this HbS is very weak. Okay, since their interaction with HbS is very weak, there is less tendency for your formation of sickle cell anemia if your HbA quantity is in high amount. Okay, heterozygote is there. In this case, you have 40% HbS, whereas your rest is HbA. So it has been seen that HbA reacts in a very less amount with your HbS to change your structure of your RBC. So more your amount of your HbA, less will be your sickling. So this is how your presence of other hemoglobin other than HbS affects your sickling of your RBC. Another one is there is another a mutation in which you get your hemoglobin C. In case of hemoglobin C, your sixth amino acid, that is your glutamic acid, is replaced by lysine. Okay, is replaced by lysine. So, but it has been seen in the presence of HbC, this HbC hemoglobin reacts with your HbS very well. So if there is higher quantity of HbC hemoglobin present in your RBC, your sickling will be more rapid, or more sickling will be seen in this case. Okay, and as well as HbF, it also reacts very less to your HbS. So if there is high amount of HbA, high amount of HbF, your sickling will be less. But HbC increases your chances of having more amount of sickling. So second is intracellular concentration of HbS. If your RBC has more amount of HbS, sickling is going to be more. So more your concentration of your HbS, more will be your sickling. Less your concentration of your HbS, less will be your sickling. Your third is your transit time for red cells through microvasculature. So in case of your transit time, through your microvasculature, it has been seen that normal transit time, okay, between your microvasculatures, your normal transit time, it is too short for it to favor polymerization. But whenever your flow of blood is very slow or your flow of blood is sluggish, it increases your sickling of your RBC. So two things: if your transit time through your microvasculature is normal, then your polymerization is going to be less, because it does not favor polymerization. Okay, whereas if your movement of your blood is very slow, or if it is sluggish through your microvasculature, it is going to increase your amount of polymerization. Okay, next is thalassemia. Okay, so thalassemia is also a hereditary disorder. Okay, in this disorder, whenever a mutation occurs which causes thalassemia, it will decrease your rate of synthesis of alpha or beta globin chain. Okay, it is a genetically transmitted disorder, so it is going to be hereditary in nature. Okay, this is also because of your mutation occurring. Okay, and in this case, whenever this mutation occurs, due to this mutation, there will be decreased synthesis of your alpha and your beta globin chain. Whenever we talk about your alpha and your beta globin chain, alpha your production of your alpha gene is generally encoded by it is encoded by 2 alpha globin gene. Wait, I'll write it. Okay, your alpha is generally encoded by, okay, this is protein, so after translation your alpha gene is being formed. So alpha is encoded by 2 alpha globin gene, which is present in your chromosome 11. Okay, whereas your beta globin chain is encoded by your single gene, which is present in your chromosome 16. So this is a pair of gene. Okay, so 2 pair means there is 4. Okay, so this is your coding, and in this case also your person may be heterozygous, which means one pair of normal allele and one pair of mutated allele for your trait, and they have a mild form of your disease, or they may be homozygous, which have both your defected pairs, and in this case they form a severe form of your thalassemia. Okay, so in case of heterozygous there is one normal and one abnormal. So in case of heterozygous they can produce normal hemoglobin synthesis can take place, whereas in case of severe form mostly transfusion is needed in this case, and this becomes evident after 6 to 9 months after birth itself. So two factors contribute to the anemia that occurs in thalassemia. Okay, two main factors normally, if you see, there is deficiency in hemoglobin due to decreased synthesis of affected genes or excess production of unaffected gene. Either there will be deficiency of hemoglobin, okay, because there is decreased synthesis of your affected gene, and there is also going to be excess production of your unaffected gene, which is quite evident. Okay, again your genes are encoded by, I already told you, 2 alpha globulin chain, which lies in your chromosome 11, while beta is encoded by a single gene located in chromosome 16, which I've already told you. Okay, so thalassemia can affect either your beta chain or globin chain, or it can affect your alpha globin chain. Depending upon the chains being affected, it may either be beta thalassemia or alpha thalassemia. Beta thalassemia is also known as a Mediterranean anemia. Okay, beta thalassemia is also known as a Mediterranean thalassemia. It is known as a Mediterranean thalassemia because it is seen in your areas near your Mediterranean Sea. That's why it is known as your Mediterranean thalassemia. Whereas your alpha thalassemia is more prevalent in your Asians than your in your Mediterranean regions. So since we are talking about beta thalassemia, it is going to result from your point mutation in your beta globin gene, which is going to affect your synthesis of your beta chain, right? So whenever we talk about mutation in case of your beta thalassemia, so this point mutation, they are different. Okay, they are more than numerous. It has been seen 100 different types of mutations are there, but majority it is a single base change is going to occur. Whenever we talk about point mutation, there is 100 different types of mutation is seen, but the most common type of mutation that is seen in case of your beta thalassemia is single base change. Okay, there is going to be change in your single base of your gene, due to which you get an altered beta globin chain. Okay, the mutation associated with beta thalassemia falls in two categories, that is B0 or B0, in which beta globin chains are no beta globin chains are produced, and B plus, where there is reduced but detectable globin synthesis. Okay, so this is more severe form of your mutation, beta not or B0, because no globin chains are produced, whereas your B plus, they have a mild form, they are your mild form of your thalassemia, beta thalassemia. Okay, so the mutations which occurs in your beta thalassemia, which can disrupt your beta globin synthesis, it normally occurs in several different ways. Okay, one is your mutation which leads to aberrant RNA splicing, and which are most common cause of your beta uh, thalassemia. Normally, in case of your RNA splicing, again they are of different types. Okay, some of these mutations disrupt the normal RNA splice junction. Okay, for your RNA to mature, right, they need to undergo splicing. So if any mutation disrupts any RNA splice junctions, it can hamper your maturation of your mRNA. So abnormal RNA splicing will lead to there will be no normal mature mRNA being formed, or in some cases your splice junctions being altered may lead to dysfunctional mRNA being produced. Okay, so when there is no mRNA being produced, there will be no formation of beta globulin. So this will lead to B naught type of thalassemia. Whereas when splicing occurs, there is numerous splicing junctions and abnormal splicing junctions is formed. It can form beta globin chain. So in this form, there will be B plus type of beta thalassemia occurring. Second is some mutations lies with the beta globin promoter and lower the rate of beta globin gene transcription. So in case of transcriptional defects, you are only affecting your rate at which your beta globin is being produced. So normally this is going to be a moderate, this type of splicing, this type of mutation will form a moderate type of thalassemia. And lastly, whenever your coding regions of beta globin chains are affected, okay, your coding regions, this is going to affect your translation of your beta globin mRNA. So change in your translation of your beta globin RNA mRNA will lead to B naught type of thalassemia, which is a severe form of thalassemia. So if translation is being affected, your proteins is not going to get formed. In this case, you are only slowing your process. Okay, in this case you are affecting your entire process of translation. So in this case no protein will be formed, whereas in this case, since you are just lowering your rate, it is beta plus, whereas in this case no protein is being formed. This is a severe form of mutation. So depending upon these three types, you get different types of thalassemia. So splicing of your junctions can give both beta plus and B naught, whereas your transcription, change in your rate of your transcription will give you beta plus, whereas change in mutation in your translation will give you beta naught. So depending upon different types of mutation, you get different types of thalassemia as well. So this is your diagram showing different changes. Okay, transcriptional defect. This is a transcriptional defect. You get your beta plus, depending upon your RNA splicing. If you see your RNA splicing, splicing of your RNA. So depending upon different types of RNA splicing, you may either get beta naught or beta plus. And lastly, there is your translational defect. So whenever there is any translational defect, you generally get beta naught type of thalassemia. So these are your translational defect. So you get beta naught thalassemia in case of your translational defects, which is a more severe form of your thalassemia. Okay, so in case of beta thalassemia, okay, there is going to be decreased synthesis of your beta globin. Correct? You have your mutation. You are getting less amount of beta globin is getting formed. Now because less amount of beta globin is getting formed, you will get inadequate formation of HbA, normal hemoglobin. You have beta thalassemia, there is decrease in your beta globin, so normally your inadequate amount of hemoglobin A is going to get formed, that is your adult hemoglobin. Side by side, okay, your beta production of your beta chain is less, but there is no alteration in your production of your alpha chain, so there is going to be more formation of your alpha chain, right? Let us consider your RBC. This is your RBC. In case of your RBC, you have your hemoglobin. Okay, in case of this, in this case, your production of your beta chain is less. So beta chain is less, but your production of your alpha chain is normal. So there will be production of more amount of alpha chain will be present in your RBC, right? So you will have excess amount of unpaired alpha globin chain. Okay, so there will be more amount of alpha chains which are not paired. So these more amount of alpha chain will not be paired because there is less amount of beta chain being produced, and this unpaired alpha chain, they normally form an insoluble aggregates. They form insoluble aggregates because they combine together and then they are insoluble in nature, so they aggregate together. This insoluble aggregates are known as your Heinz body, H-E-I-N-Z, Heinz body. And these insoluble aggregates, they precipitate in your RBC. So this precipitation will go and damage your membrane of your RBC. So due to which your lifespan of your RBC decreases. Again, I'll repeat. Okay, beta thalassemia is going to affect the synthesis of your beta chain. Okay, so let's draw your RBC. So this is your RBC. So you are suffering from beta thalassemia. Your beta chain is going to get produced at low amount. Side by side, your alpha is going to get produced at your normal rate. So 4 beta that has been produced will go and bind with 4 and then form a normal hemoglobin, whereas rest alpha, they are unpaired. So this unpaired alpha chains, they normally form an insoluble aggregates, which is known as your Heinz body, and this insoluble aggregates, they go and they precipitate in your RBC. This precipitate it goes and it is going to damage your membrane of your RBC. So your damaging of your membrane is going to lead to reduced lifespan of your RBC, correct? So there is going to be extravascular hemolysis taking place. So whenever there is hemolysis, degradation of more amount of your RBC, anemia. So if there is anemia, your blood carrying capacity, your oxygen carrying capacity of your RBC decreases. So hypoxia, erythropoietin is going to increase, bone marrow expansion, due to which you get your skeletal deformities as well. Other than that, whenever you are destroying more amount of your blood is getting destroyed, there's going to be iron overload as well, because first and foremost you are eating iron from your diet, which is getting absorbed. On top of that, you are getting more amount of iron, which is normally recycled. Whenever your blood is getting destroyed, you will have more amount of iron accumulating inside your body as well. So there will be iron overload taking place side by side. Okay, so next is your alpha thalassemia. So when we talk about alpha thalassemia, in case of alpha thalassemia, there is a deletion of your gene which results in defective alpha gene synthesis. Okay, deletion of any amino acid sequence of or amino acid base pairs from your gene will lead to alpha thalassemia, and due to which there is going to be a defective alpha gene synthesis. Okay, so in case of your alpha thalassemia, it has been seen that more your amount of deletion, more your signs and more severe your thalassemia is going to be. So whenever only one gene or a single deletion is there, you get a asymptomatic alpha thalassemia. You do not get any symptoms of your thalassemia, and it is just a carrier. It does not have any harmful effect on your body. Whereas whenever there is two gene deletion, I said two genes are present which code. So there are two pairs of genes which code for your alpha globulin chain. So if there is deletion of a single alpha globin gene, they are asymptomatics. They are only carriers. They do not show any symptoms of anemia at all. So whenever there is two gene deletion, you get your alpha thalassemia trait. They carry your trait and they exhibit mild hemolytic anemia. Whereas deletion of 3 or 4 chain genes will lead to a severe form of your alpha thalassemia. Okay, you get a severe form and then it is clearly seen as well. Okay, and this normally leads to your unstable aggregate of chains of hemoglobin called HbH. And alpha thalassemia is more common in your Asians. Okay, next is your megaloblastic anemia. Normally megaloblastic anemia is caused by impaired DNA synthesis, okay, due to which, or you can say not just synthesis as well as replication, okay, impaired DNA synthesis that results in enlarged red cells due to impaired maturation and diffusion. Two main causes is vitamin B12 deficiency and folic acid deficiency. Both vitamin B12 and folic acid, they play an important role in your synthesis of your DNA. So if if anyone is deficient of vitamin B12 as well as a folic acid, then it will lead to megaloblastic anemia. Okay, so first we'll talk about your folic acid deficiency anemia. So normally folate or your folic acid, it is found in all food, okay, food items, okay, all your food items, they contain folate or your folic acid. So whenever we talk about folic acid is normally stored, okay, in your body. It is stored near around 2000 to 5000 microgram of your folic acid is stored in your body, and daily requirement of your folic acid is around 50 microgram only. You already have your storage of 2000 to 5000 microgram, and daily requirement is around 50 microgram only. So in case, even if you have a dietary deficiency, for you to, okay, there is a dietary deficiency of folic acid, for you to develop anemia, it is going to take few months. Okay, only after being deficient of folic acid for few months, only then will you see your symptoms of your anemia. And your most common cause and your most common causes for folic acid deficiency are malnutrition in dietary or lack, especially in elderly or in association with alcoholism. So all these things can cause to for your deficiency in folic acid. So normally folic acid helps in your synthesis of your purines and thymidinates, okay, which we'll talk about in a while. Second is your vitamin B12 deficiency anemia. Okay, vitamin B12 is also required for DNA synthesis and nuclear maturation, okay, and which in turn leads to normal red cell maturation and division as well. Vitamin B12 is found in, it is also found from your food source, it is obtained from your food source in foods of all animal origin. Generally dietary deficiency is very rare, okay, it is very rare to see anyone deficient of vitamin B12, and it is usually seen in strict vegetarians who avoid all dairy products as well as meat and fish, okay. Otherwise it is very rare to see a person deficient from vitamin B12. So since vitamin B12 is found in your food source and normally from all your biggest food source, whenever you take vitamin B12, okay, whenever you take vitamin B12 from your food, it is normally bound with, your vitamin B12 is bound to a protein, okay. So when it reaches your stomach, your pepsin acts on your food source, it cleaves off your protein from your vitamin B12, and then you get your, okay, normally your vitamin B12 is not in your free form in your food, it is bound to a protein. So after you take your food, in your stomach, pepsin is going to act in this complex, it breaks off your vitamin B12 from your protein, you get your separate vitamin B12. Okay, after it gets separated from your vitamin B12, okay, it combines with a salivary protein which is known as your haptocorrin. When pepsin acts and liberates your vitamin B12 from other protein, it normally binds, when you swallow your food, your salivary protein also enters. So after it has been separated from your protein, it goes and it binds to a protein which is your haptocorrin, which is normally your salivary protein. So this complex of vitamin B12 and your heptachlorine, it goes and it enters into your duodenum. Okay, it goes and it enters into your duodenum, and in your duodenum, okay, so right now you have your vitamin B12 and your heptachlorine complex which has entered into your duodenum. In your duodenum, what happens? There is action on this protein by pancreatic pancreatic proteases. Okay, so your pancreatic proteases is going to act on this complex, that is your vitamin B12 and your heptachlorine complex. This pancreatic proteases, what they do, they are going to break off this bond between them, so you get your separate vitamin B12. Now again, after it has reached your duodenum, pancreatic proteases is acting, breaking off your bond between your vitamin B12 and your heptachlorine. Next, this vitamin B12 is going to go and bind to your intrinsic factor. Okay, this intrinsic factor is produced by your parietal cells present in your stomach. Okay, now this complex, vitamin B12 and your intrinsic factor complex, it goes and it binds to a receptor known as your tubulin. It goes and binds to your receptor known as tubulin, which is a receptor for your intrinsic factor. It binds there and it is taken inside your cell. It enters into your cell. So after it enters into your cell, your vitamin B12, it goes and it binds to your transcobalamin. Okay, it binds to your transcobalamin, and then this complex is transported throughout your body. Wherever it is needed, it is taken and then used. I'll repeat it once again. Normally your vitamin B12, this is found from your animal source. So on your time of ingestion, okay, it is already bound to your animal protein. So when it enters into your stomach, it gets cleaved off, and then you get your separate vitamin B12. To this vitamin B12 binds your salivary protein, which is your heptachlorine. So this heptachlorine enters into your duodenum. In your duodenum, your pancreatic protease is going to act and then break off this vitamin B12 and heptachlorine complex. So now you have only vitamin B12 in your duodenum. So to this vitamin B12 comes and binds your intrinsic factor, which is produced by your parietal cell. So this complex goes and binds to your receptor tubulin, to your tubulin receptor, which is your receptor for your intrinsic factor, and then vitamin B12 enters into your cell. That is, it enters into your cell in your ileum. After it enters into your cell, it goes and it binds with your transcobalamin. So you get your complex vitamin B12 transcobalamin 2 complex is there, and it is transported throughout your body. Okay, so this is your normal. This is how it is normally taken inside your body. So main function of your vitamin B12 in case of your formation of your blood, okay, is just one is your transfer of your methyl transfer. In this case, one important function of your vitamin B12 is to transfer a methyl group from, okay, it is going to transfer one methyl group from N-methyl tetrahydrofolate, okay, to homocysteine. So from, see, if you see this entire this okay, see this entire thing, what is happening is your cobalamin, which is vitamin B12, it takes up a methyl group from N5-methyl tetrahydrofolate. Okay, so after taking one methyl group, it gets converted into methyl cobalamin. Okay, on doing so, it is forming tetrahydrofolate, which is very much important, okay, for production of your DNA. And then it transfers one methyl group, this methyl group, to your homocysteine to form methionine. So main function of your vitamin B12 is to transfer methyl group from your methyl tetrahydrofolate to hyoscine to produce methionine, and side by side it forms one important compound, that is your tetrahydrofolate, which is very much important for your formation of your DNA. Okay, so if this, okay, we'll go to the next slide first. One of your important cause of vitamin B12 deficiency is pernicious anemia. Pernicious anemia, this pernicious anemia is a autoimmune disorder. Okay, it is an autoimmune disorder, and this autoimmune disorder goes and it affects your, destroys your own body's parietal cell. So if your parietal cells is getting destroyed, intrinsic factor will not be produced. If intrinsic factor will not be produced, your vitamin B12 will not be absorbed. Okay, so pernicious anemia is an autoimmune disorder, in which your own body's antibody is going to go and destroy your own cells. So in this case, your cells in target is your parietal cells, so they go and destroy your parietal cells. If your parietal cells are getting destroyed, intrinsic factor is not going to get produced. If intrinsic factor is not being produced, then absorption of your vitamin B12 is not going to take place. Okay, and chronic malabsorption is also seen after gastrectomy or ileal resection, and there is a correlationship between your metabolic defects between your vitamin B12 and your folate metabolism as well, because vitamin B12 is required for recycling of tetrahydrofolate to the form of folate that is needed for DNA synthesis. Okay, so we'll see it. So this entire thing, this entire chart explains your need of your folic acid or your folate, as well as vitamin B12 for your formation of your RBC, okay, or for your formation of normal DNA synthesis. So first and foremost, I hope you did not forget this. Please note over here, N5 methyl tetrahydrofluorate. This is, I said folic, wait, okay, folic acid, right? I said folic acid is formed in any type of food that you eat, you get your folic acid. So the folic acid, your dietary folic acid, is basically N5 methyl tetrafolate. This is what you get from your diet, as well as this is the form in which your folic acid is stored as well. Okay, it is N5 methyl tetrafolate is what you get from your diet. It is how it is stored in your body as well. So over here, so first see this one, N5 methyl tetrafluorate, okay, is a dietary fluorate, as well as from your storage it needs to be converted into tetrahydrofluorate. And one of your most important mechanism by which it is converted is with the help of your vitamin B12. Okay, so tetrahydrofluorate you get second important thing that we have to take into consideration of your folic acid is folic acid with the help of your fluoride reductase is converted into dihydrofluorate, and dihydrofluorate is converted into tetrahydrofluorate by fluoride reductase, both of which is going to form tetrahydrofluorate. Tetrahydrofluorate is one of your most important component for your formation of your purine and your thymidylase, which is responsible for formation of your DNA. Okay, so if there is any deficiency of vitamin B12, as well as if there is deficiency of folic acid, there will be no formation of tetrahydrofluorate. So if there is no formation of tetrahydrofluorate, there will be no thymidylate synthesis taking place. So if thymidylate synthesis does not take place, your synthesis of your DNA is disrupted, as well as your purine synthesis. If you see over here in your top, purine synthesis does not take place. So any deficiency of this is going to disrupt your normal DNA synthesis. So if your DNA synthesis disrupts, there will be no normal maturation of your nucleus. There will be no normal production of your RBC as well. So well matured RBC is not produced, because of which you again get megaloblastic anemia. Next type of anemia is iron deficiency anemia. Iron deficiency anemia is the most common type of anemia that you generally see throughout your world. It is more common in your developing nations than in case of your developed nations, and it results from dietary deficiency. Okay, your iron deficiency anemia or IDA, either there is dietary deficiency or loss of iron through your bleeding or increased demands. Any one out of these three causes can lead to your iron deficiency anemia. Okay, so normally your source of your iron is also again from your food. Okay, food is your main source. So iron balance is maintained largely by regulating your absorption of dietary iron. Also, your iron is generally recycled in your body. Okay, so it is not only through your diet but also through your recycling of your iron that takes place in your body itself. So iron is a component of your heme. Okay, main component of your heme. We already told you, heme center there is an atom of iron. So if you have less amount of iron, there will be decreased hemoglobin synthesis. Okay, iron in the form of hemoglobin is constant. Okay, I told you, see in this case, important attribute of iron is that it can be recycled. Okay, so because of this recycling taking place, your body can balance between your iron that is in use as hemoglobin and iron that is stored and is available for future hemoglobin synthesis as well. So there is storage and it is always recycled. So see, if this is your erythrocytes, when they go and they get destroyed in spleen, liver, right, what happens is your globulin part, protein part, it goes and it gets reabsorbed or it is thrown away, whereas your heme portion, your iron portion, it enters into your blood and then it is transferred to different parts of your body, stored in your liver, stored in your spleen, and whenever it is required, it is released and then it goes into your red bone marrow, and then there is new production of erythrocytes. So it is always maintained, it is recycled. So generally a very less amount of iron is lost from your body. Okay, so your iron deficiency anemia, it generally develops slowly. Okay, so there are three overlapping stages: stage 1, stage 2, and stage 3. In case of stage 1, normally the body iron stores for RBC production is used and hemoglobin synthesis are depleted. But in this case, normal RBC production is being done because your stores, your iron stores that is there in your body is giving out iron for your production of your RBC. Okay, so there is going to be depletion in your storage. Second is stage 2. There is insufficient iron amount of iron are transported to the bone marrow. In the first stage, you are depleting your stores. Though you are depleting your stores, since you are depleting your stores, iron is still present. So normal production is going to take place in stage 1. In case of stage 2, now you have already depleted your stores. So since there is depletion of stores, your iron being transported to your bone marrow is going to be less. So you may be producing hemoglobin which are normally deficient of your iron, because you have already depleted your source, you are not getting more amount of iron, so your hemoglobin being produced are normally deficient of your iron. And lastly is your stage 3. Okay, stage 3 is when the hemoglobin deficient RBC enter into your circulation, and this is your stage in which your manifestation of your anemia or iron deficiency anemia is seen. So 3 stages. In stage 1 you are depleting your source or your storage. In stage 2, since you are depleting your stores, you get less amount of RBC, less amount of iron is going into your bone marrow, and hemoglobin is being produced. Now they have less amount of ions in the hemoglobin. Iron deficiency hemoglobins are being produced. And in case of your stage 3, when it begins completely, your RBC, they do not contain any hemoglobin. So if they do, due to which you get your manifestation of your anemia in your stage 3. Lastly, we have your hemophilia. Okay, hemophilia is not related to your anemia. Okay, hemophilia is a disorder which affects your clotting of your blood. It affects your coagulation of your blood, or you can say your clotting of your blood, and it is generally inherited. Okay, it is passed on from your parents to your offspring. So there are two types of hemophilia. There is hemophilia A and there is hemophilia B. Okay, so first we are going to talk about hemophilia A. Hemophilia A is one of your most common type of hemophilia. It is X-linked recessive disorder. Okay, it is an X-linked recessive disorder, and because of this disorder there is basically decreased factor 8 activity. Okay, factor 8, which is a factor needed for coagulation, there is decreased amount of factor 8 activity. Either they are not being produced more, or their quality is not good enough to produce coagulation. So there is decreased amount of factor 8 activity, and generally this affects your male more than your females, whereas females are just your carriers. Though it is hereditary in nature, it has been seen that 30% of new diagnosed cases, they do not have any family history. So new mutations can also lead to formation of your hemophilia A. So I've told you it is an inherited disease, which has to be passed on from your parents to offspring. But it has been seen that 30% of new cases, diagnosed cases, they do not have any family history of this disease. So a new mutations can also lead to hemophilia A. Okay, so whenever we talk about this, I said uh, out of all your hemophilia types, 90% of your hemophilia patients of your hemophilia A, they generally there is insufficient quality. Okay, there is insufficient, sorry, not quality, insufficient quantity. There is less quantity of factor 8. That is, factor 8 is being produced at a less amount, whereas in case of 10% of your patient, it has been seen that a defective factor 8 has been formed. 90% insufficient quality, quantity, insufficient quantity, which means less amount of factor 8 being produced, and whereas 10% of patient, a defective factor 8 is being produced. So okay, so the percentage of normal factor 8 activity in circulation depends on genetic defect and determines the severity of hemophilia. That means more severe your genetic defect, more severe will be your hemophilia occurring. So if your hemophilia is mild or moderate forms of your disease, bleeding usually does not occur unless there is local lesions or trauma. Until and unless there is no damage, you do not see your bleeding occurring. In case of severe hemophilia, okay, if you are suffering from severe hemophilia, bleeding usually occurs in childhood and is some spontaneous and severe, often occurring several times a month. So this is also again depending upon whether you are homozygote or heterozygote. Heterozygote, you have a mild or moderate form of disease, whereas a homozygote, they are going to have severe form of disease. So with this, we end the chapter. Thank you.